Hi everyone, thanks for joining us at the second webinar of the FAIRDom Data and Model Management webinar series. I'm Natalie Stanford, the FAIRDom Community Lead, and I'll be your host today. FAIRDom is an initiative to support researchers, students, trainers, funders and publishers, in fact anyone interested in data and model management, in better data and model management practices. We offer software, advice, expertise and training, which make managing your data and models in life sciences easier. You can find more about FAIRDom at our website at www.fair-dom.org, or you can follow us on Twitter at FAIRDom_EU. If you have any questions regarding FAIRDom, you can get in touch with me at community@fair-dom.org. Now, today we're here with our guest, Brett Olivier, and he's from the VU in Amsterdam in the Netherlands. Morning, Brett. Brett?
Yep, good morning,
Hello?
Natalie. Hi, sorry about that. Was still muted.
Okay, how are you today?
I'm well, thank you. It's a great day here in Amsterdam, and welcome to everybody that's joining in this webinar. Absolutely, it's a pleasure to be here.
It's good to have you here. Now, Brett trained as a biochemist in the Triple J group at Stellenbosch University in South Africa, and during his PhD and postdoc, he specialised in analysis methods and software development for systems biology, and he generated the software Pisces and JWS Online. He moved to the Netherlands to work on broadening applications for his work and standards that assist with his work, and it's this standardisation work that he's going to be talking about today. If you've got any questions during the session, you can raise your hand, which should be in a panel on the right-hand side, and we'll then select you at the end for some questions, or you can also post your question in the chat and we can select these questions to read out to Brett once he's finished his presentation. Just a reminder that the webinar is recorded and we'll make it available on our website in due course. So I'm just going to hand over to you now, Brett.
Hi, yeah, thanks. I'm just trying to, the only problem I have at the moment is that I'm sharing the screen.
You should have.
Yes, that's much
There we
better. There we go. Yeah, does that look okay?
It looks great.
Okay, excellent. So it's a great pleasure to be able to give this presentation here. And so, as Natalie mentioned, I'm involved in software and standards and also modeling. And so today I'm going to be looking at modeling and its implementation and standards, especially then see how this can be used to make data more fair. So the talk is going to have three major sections. My apologies to Sergio Leone, but it will be looking at how things were, which wasn't so good, what you can do about it, and how things should be in the future. What I'm going to be talking about, the context is going to be systems biology and modeling in systems biology. And to most people, if you ask what is systems biology, you get some other thing about a cycle. It has models. It seems incredibly complex. But in essence, if you reduce it down, you can simplify this down to some sort of methodology where you have an interactive cycle between experimentation, modeling, and theory. And this is the modeling context that I'm going to be talking about. And the models that you use. What is usually missing from these cycles is something that is going to be something that I work in, which is relatively important, and that's some computational stuff, which sort of which underlies this entire cycle. So if we look at the model itself, we're talking about fair data. If we talk about fair models, the point that I'm going to be trying to make is that to have fair models, we need to have standardization and we need standards for model encoding implemented in software. This shouldn't be so strange because everything we do these days depends on standards. Whether you're talking on your cell phone or whether one cell phone is talking to another cell phone, this depends on standards. However, the application of these standards to mathematical modeling or quantitative modeling is relatively new, and we can say this has been the last 10 or 15 years where this has actually become an issue. If you think about models themselves, and here we talk about quantitative models in systems biology, of course, these themselves are actually data. They're exchanged between researchers. And more are being used in ways that wasn't originally thought about by the original authors. And so as soon as you talk about interchange or exchange, then the questions come up that are captured in the whole FED initiative. How big do they become accessible or interoperable? The interesting thing with models themselves is because they're almost executable, they can quantify them, you can computationally use them to produce a result that go beyond, actually, the question goes beyond exchange, but actually to interoperability, in that you can start saying, how do we get these tools to talk to each other through the use of standards? So to narrow things down a little bit, let me start talking about what is a model, and what is the model that I'm going to use. And I've wanted the example I'm going to use today is going to be constraint-based modeling or genome scale modeling. This is a very popular way of doing things at the moment. It has a wide application both in basic research as well as applied research in everything from community analysis to drug design and stuff like that. So it's relatively interesting. It has a nice story behind it, in that it essentially is a way of building a model from information that we have, which is nowadays relatively popular or commonplace, and that's genomes. So from the genome, if you have a sequence genome of an organism, you can then annotate this genome through a process of homology, and from the genome you can convert it essentially into a large metabolic network of reactions. Once you have the metabolic network of reactions, you can then say, okay, this is now, we know what substrates are, we know what the products are, and we have stoichiometry. Once we have this network, we can add quantitative information to it, such as bounds on the fluxes or an objective function, and essentially we can redefine this as a linear program or an optimization problem. So that's the model. When I talk about a model today, it's going to be generally like a combination of reactions, metabolites, compartments, genes, fluxes, bounds on fluxes that can be optimized. Now, while the actual model description seems relatively simple, the problem comes in here with the scale. Okay? Because while it all looks very neat and simple, and this really nice example of mine here, what you have in real life is something really large. We're talking about a whole cellular metabolism, and this can have thousands of reactions and metabolites. And all of these things have to be identified and worked with, and transferred or exchanged between people. So the core question at the moment is then, okay, if you have one of these models, how do we describe it? And then the future question is, how do we describe it in a way that we can actually use it and exchange it, right? So if we go back a few years and we look at what the situation was, say, in 2009, I'll get back to why I chose that particular date, then the problem was quite bad. The situation was like this. Each model could be defined in using a different format, and everything from a PDF to an Excel spreadsheet to a text file to a Word document, some sort of arbitrary XML, and also a dialect of a known language called SPML that was used by a specific tool. Okay? And so this was a tool-specific dialect, which was, in the end of the day, the best way of describing things out of all the other options, but it had led to some interesting consequences. And that's what I'd like to explore now, is to look at the dangers, let's say, of misusing a standard to start off with. So you might say, well, this situation is fine. Who cares? Only a few people are using it. But it actually turns out that this has real consequences that can mean time and money in the end. So the Cobra toolbox is like was at that stage and has been for a long time the de facto standard for encoding constraint-based models. And it used this Cobra dialect as its own input language. It's well supported, well respected piece of software, and that was all fine until in 2014 there was this paper was published that came along and said, well, there's a problem. First of all said that you couldn't do flux balance analysis with rational arithmetic, something that could be done by Cobra and every other tool up to that point. And then it said that most of the models were wrong, or at least 50% of the models didn't work as they were supposed to. And this was quite a major thing, because on the one hand you have an established way of doing things, and communities use it as backing, and suddenly the group comes along with a completely different result. Now, there are many issues around this particular case, but I'd like to just pick up on one. As it turns out, there was nothing wrong with actually using rational for certain things. You may need exact arithmetic, but not for general FBA. So where did the problem come from? And it turns out the problem wasn't actually with the method at all, but it was with the models themselves. And the problem came in the actual interpretation of the model. So say we had one model described in what was then the SBML that these tools supposedly understood. What would happen is you could take this single file and you could read it into both different tools, and you get a different result. Now, this is a little bit strange, because if you think of it, you may have a standard that's supposed to be able to do this, but it was really, the problem came from the fact that the original Cobra dialect was not SBML, and so there was no reason for the Mongoose group, or the group that developed Mongoose, to decide to use it in a different way. And this is just an example of some of the things that happened. I mean, if you look at the first one, let's say which metabolites will be free or fixed. I mean, the Mongoose tool decided that it was all of them in a specific compartment. The Cobra approach would depend which version you were using of Cobra. In early versions, it depended if the name ended in an _b, or in the later cases, using the actual attribute that SBML has for this, which is boundary condition. The other cases are all the same. It literally just takes something that exists in the language, use it in a tool-specific way, and then say that everybody else that doesn't use it your way, or doesn't interpret it in your way, is incorrect. So the question is, how did this come about? I mean, this is a crazy situation, and none of them are correct at that point in time. They were both not. If you read both of these in an SBML tool that was actually specifying SBML correctly, then it would get yet another interpretation of the model. So let's say it's not the standard itself, it's actually how the standard was implemented, right? Now, this is a sort of the history of what went on. And the important thing here is it's not really important how it happened, because I have no idea why these things were, but this is sort of how it turned out. In 2005, when Cobra was developed and when SBML was still very young and not widely used, they started using their own dialect. Okay, this was fine. There's nothing wrong with that at the time. But then Cobra develops, and as the fact that it's the de facto tool, it becomes the standard for usage in the constraint-based modeling community, and therefore this dialect becomes the standard way of encoding a model. that I know of, there was never really a specification document that said this is exactly how we use it or what it is. It was just simply what worked with Cobra or what didn't. And this, of course, could change between different versions. Now, if we look at this, what was happening at the same time in the SPML community, it was actually mentioned in 2006 by Karthik Raman at a meeting in Tokyo that there was this issue that people were using it like this and that maybe SPML should do something about it. Nothing happened, unfortunately, at the time, until 2009, when I started working with constraint-based modeling and developing using these models and getting totally frustrated that everything was encoded in a different way, and that I was writing an adapter for every separate model that was out there to parse it. I opened up and said, well, maybe we should do something about it in SPML. I've been working in SPML for a while before that. So everybody thought this might be a good idea to proceed with at the time. What helped a lot was in 2010, when SPML level 3 was specified. And I'll get into more, I'll show in more detail what the details of SPML are and what this actually means. SPML 3 was released, which allows the language to be extended. And this meant that we could go ahead, and myself together with Frank Bergman, started working on an official process for specifying an SPML package extension. This, of course, takes a lot of time. It involves much discussion with the SPML community. Once you have a proposal, it means the formation also of a package working group. And getting more people involved, and through a process of a few years, eventually resulted in 2013 us releasing the first version of what is called the FPC package, which is an extension for SPML to handle these constraint-based models. Just soon, relatively soon after we'd released this, actually the Mongoose thing happened. And as positive out of the whole scene is that it meant that a lot of people suddenly noticed that there was this work taking place and got involved. And then after another relatively short period of time, and with much input from a lot of people, so both from the SPML communities and from the Cobra developers and constraint-based modeling teams, we finally end up with a package specification that everybody uses or is happy with using. And so this is great. So what is the moral of the story? Well, while it doesn't take very long to make up your own version of a standard, it can take 10 years or so to fix it. So, if you're going to use a standard, please use it correctly, but the real moral of the story is get involved. If you feel that your modeling thing is modeling type or methodology is not represented, you can go and make up your own. You can also get involved and make the standard work for you. The advantage, of course, is that once there is a standard that everybody supports, there's now more and more tools being developed that use it. Cobra, the big databases. There are now converters being made, and a lot of tools, and now there's suddenly interoperability. So if you look at the FAIR principles, interoperability and reproducibility are suddenly way more enhanced, just from the fact that now we have a unified standard. So that's an example of how things can go wrong if you don't use a standard properly. Let's look at some of the more techniques. I've been talking about SPML. So let me go into a little bit more detail now for a few minutes about what exactly SPML is. SPML is a markup language that is used to encode biological processes or models. It actually forms a key part in a family of different standards in language, so it allows support for Miriam, which is an annotation, minimum annotation standard. It makes use of biomodels qualifiers, which can be used to in RDF statements, for example. It supports its own ontology, the system biology ontology, and it allows for proper annotation. So this is great. It forms actually sort of like a hub for a lot of others, connecting together a lot of standards. If you look at it in more detail, essentially it's process description. Something is acted upon and creates something else. It happens to use a semantic which says that a species goes into reaction and comes out as some other species, but what these species are can be anything. It's not necessarily only a biochemical species. This description is quantitative, the fact that it actually has explicit mathematics. And so this is essentially what makes up SPML. So you can describe a model in terms of processes, reactions, metabolites, things like parameters, compartments, time-dependent events. Now, SPML level 2 was essentially this, the core language. SPML level 3 took this and said, well, this is SPML core, and we can now extend it using different what is known as packages. So for your example, here we have a model which uses SPML level 3 core as its base description, but it adds on to that things to do with constraint-based modeling. So example, the FPC package, or uses packages called hierarchical composition for adding different models together into a single model. So these are the packages which have been approved. There's packages for qualitative models, layout, the FPC ones, and there are a lot more in development as well. There are packages for arrays and all this type of thing. These are available also on the SPML website. So SPML is trying to allow extend its range in the type of models that can be encoded. The big advantage of using SPML is actually not just the language itself, because otherwise that would just be a single document. The big advantage is that if you're using SPML, you get an entire community working with you. There are libraries for using SPML, for interpreting it, for example, libSPML, which has APIs for a whole wide range of languages. There's JSPML, which is a native Java language library. There's also an entire community which has with support for discussing things like interoperability. Plus, you get a whole lot of tools which are developed as part of the SPML core team. Things like online validation and converters between different levels and versions of the SPML itself. So there are also things like software comparisons and software guides for using it. So that's SPML. Another language which is related to SPML is also called ZML. ZML is the simulation experiment description markup language. And ZML implements a minimal standard description called Miyazi, which is to basically describe how to do a simulation. Yeah, you can think of something like a time simulation. So as an example, whereas before we have a model description, it's an SPML model with all its associated detail. ZML allows you to take a simulation, which is simulation consists of something like an algorithm, what type of simulation it is, how it starts and ends, when it starts and ends, and combines these things together into something called a task, which then generates data, and that can be processed in different ways to then presented as an output type as a report. So in other words, what this is saying is take model X, set these parameters, run it for 10 minutes using LSODA, and plot these results. So now not only the model, but also the experiment you define. So this has only been two of standards that I've now briefly mentioned. There are a lot more. CellML is another widely used standard. There's both for synthetic biology, languages that are specifically for graphical notation and stuff like that. And all of these are available from the Combine initiative, which is sort of a home online for these type of standards. I invite you all to take a look there to get hold of the latest specifications and details. So that was diary detail out the way. Wasn't so bad, I don't think. So let's look at what could be the good. And the good is going to end up on the way I'm going to say, well, okay, let's take what is a day in the life of a computational biologist. Imagine that you come into work, you're a computational biologist, and somebody says to you, well, today you want to make a model of yeast glycolysis in a growing cell. You want to do this both on a detailed level and on a whole cell level. So how do you go about it? Well, fortunately, first thing to do is to jump onto Google or somewhere, some resources. So you want the details of yeast models, glycolysis, you want a detailed model. You know that there's a detailed model of SPML repository of models, and this is something like biomodels. So you go and search in biomodels, and there you find the classic, let's say, model of yeast glycolysis by Bostosic. You decide this one has what you want in it. The annotation is clear from the database, so the things, it has the components you want in, and the literature says that this seems to be a good model. The first thing you might want to do is load it into a program like Copasi, which is for systems biology simulator. And then there you can see again exactly what is in it, play around with the parameters, run some simulations, and check whether it's actually the model that you want it to be. This is great. At the same time, you think, well, you didn't want to only work on a micro scale, you want to work on a whole cell scale. So for that, you need a different type of model. So you need a whole cell model of yeast, and you've heard that one exists. A community consensus model is available from the site YeastNet on SourceForge. And you think, well, that might be also interesting. So fortunately, you can say you can download that model in SPML level 3 and load it up into the FAME online simulator. This is software that we've developed for doing online simulations, online FBA basically. So it's also nice to explore. You can load the model, see what's inside, run it, see it. And that's great. So now you've thought, well, you have these two models, you can set them up to be like you want, but you'd like to go a little bit further than this. Instead of dealing with these individually, you'd like to combine them in some arbitrary sort of analysis, right? So for that, you need a slightly different type of tool. And if it was myself, I would be using these two, which is software I happen to develop, one of them called Pisces, which is the Python simulator for cellular systems. This is a kinetic simulator. It's a Python library type thing, interactive Python thing. And it is related, has a related piece of software called CBMPy, which is for constraint-based modeling. So through some nifty sift, and both of them understand SPML, although the V1 seems to have disappeared, but both of them can read SPML, so you can take your files that you've modified in the software, put them together, combine them together in a new way, and do your analysis, right? So now this is not only data integration, this is now interoperability, and it actually allows these simulators to be combined because the model descriptions are compatible. And that comes from use of a standard to encode them. Right? Of course, once you've done it, you've got a brilliant result of something, now you want to store it. And there's one standard that I haven't talked about yet, which is very useful, and that's the Combine Archive. Right. So with the Combine Archive, it's essentially a zip file with the metadata, and what you can do is then you can combine, for example, ZML scripts which describe the simulations that you did previously, the SPML models themselves, any links to outside data, so for example PDFs or metadata, you can put that together, and that uses a format called OMEX, so Open Modeling Exchange, and so you can then export from this combined software, you can export a sort of a combined experiment, right? And store these all together so that you can then email that off to whoever asked you the question initially and say, look, here is my simulation, the models and everything, here's the results of the experiment that you wanted. Right? So that's how it should be. It's not like that yet. There are lots of other issues, but that's how it should be. So the take-home messages of this that I'd like to emphasize now are, even though it's not a very popular topic, standards are essential. And it's not just as an esoteric essential, it's something that computer people also deal with for software. It can really affect how your work is done and how your workflows are set up. It's no use having standards if they're not implemented in software. You know, it sort of sounds obvious, but it's very true. And if you're going to implement software, stuff in software, even if you implement a really small part of the standard, at least support it, implement it properly. You know, as I showed earlier, forking a standard can be very costly in terms of time. So nobody says every software has to support every standard, but at least the bits that you do right. And to do this is critical that you engage, whether you're modeling or whether you're developing software or whether you're creating models. Engage with standards communities. Especially if it doesn't do what you want, because in the end, you are the domain expert. Standards people are relying on people to come to them and say, this is a problem, how can we fix it? And of course, as in everything, do not reinvent the wheel. So as in any community, it thrives on new people and new ideas. So finally, to end off with, I'd just like to acknowledge the people that I've worked with here, and people in the SPML, ZML communities, and these are the funding people that support me in this sort of research. So great. If there are any questions or anything, I suppose Natalie sort of will take over now.
Yeah, thanks very much, Brett. That was a really interesting talk. Yeah, if anyone's got any questions, if you can raise your hand, I'll come to you in a bit. I'm going to take the liberty of kicking off with the first question, though. So what I found quite interesting about this was that the main problem was that you had a standard defined, but people were using variables that you could label yourself freely as an official syntax for programming. Is that right?
Yeah, essentially there were, if you look at the details of it, what was happening was they were overloading certain attributes, so they were giving semantic meaning to to for example, identifiers that only they understood.
Yeah, and so you proposed at the end this idealized pipeline of how you think we should be working, and it obviously involves standards at each step to try and negate things like this. But do you think that pipeline itself will inherently remove the problems that occurred, so they wouldn't happen again, or do you still think there's a chance that without proper implementation it could happen again?
Well, there's two levels of access in the pipeline on the thing. In terms of the standard development itself, that can be avoided by using an inclusive process and saying, well, if we want to use a standard, we try and change the standard to handle other people, all cases. So that's the one thing. Instead of creating your own version, you try and extend the version that exists. That's on one level. But on the other level, as far as where there is really a lot of work that's needed now is, of course, something that I missed out completely in the pipeline, and that is that, for example, we might have two models of yeast where the identifiers are talking about the same thing, but they are completely different.
Yeah.
And the annotation is patchy and inconsistent, and therefore you can't do that. And that's exactly the problem that exists there, and where something like fair data or fair models make a lot of sense.
Okay. We've got one hand up at the moment, which is from Carol Goebel, so I'm just going to unmute Carol and see what Carol has to say. Hello, Carol?
No, I'm sorry, that was a mistake.
Okay.
I don't have my hand up.
Okay,
sorry.
well, okay, it's okay then. Does anyone else have any questions? If you just look at your right-hand panel, you have a hand you can raise, or you can ask questions in the questions box below or in the chat. So far, you're all very quiet today. So I think, Brett, we might have some shy people on our webinar today. So what I'm going to suggest is we actually have a forum. So if you go to our website, if you go to the handouts at the bottom, you can download the contact details for Fairdom. And you can go to our website through a link on the PDF. If you go to community and go to BioStars, you can actually post some questions in the BioStar forum. And Brett's already agreed before the webinar that if you have any questions after the webinar, he's very happy to go there and answer them. Isn't that right, Brett?
Yeah, absolutely.
Yeah. Okay. Because as I say, it was a bit more specialized today with specifically SBML, but I think the actual underlying principle behind it of using standards properly and people really needing to get involved is the main take-home message. Carol's decided that she wants to ask a question now anyway. So let me just see. You've got a question now, Carol.
Yes, I thought, why not? We can't let Brett disappear without a question. That would be unacceptable. Brett, so you eloquently put these standards fork, and people tend to go off into their own enclave and evolve their own version of the standard, as it were, rather than kind of participating as part of the mainstream trunk of the standard. So with the benefit of hindsight, what are the kind of environments that we can do how can we create an environment in a standardization activity to avoid that happening? So that particularly, I wonder, because we have a cost action just starting in Europe on standardization, trying to bring standardization in life sciences together. It's called the CHARM action. And how could we go about setting up that action so that this kind of disappearing off into a corner with your own cronies is avoided?
Yeah, this is a very interesting question, and it actually goes on to something that I've started thinking about. Is that once you have a encoding standard, for example, that's really only the start. especially in terms of models where you say, well, okay, now I have an encoding standard, does that make a good model or not? So it comes more into a quality control sort of thing. Can you set the quality of a model? And if you start looking at it from this perspective, then it becomes a lot more interesting, because then you need a lot of people from the different domains, from the experimental to the modeling to the standards, to work together to be able to answer those sort of questions. So I think it starts with, on the lowest level, it starts with software developers. Of course, there is always the problem that the tool developers, it takes a lot of time, and it, in scientific terms, wastes a lot of time to implement these standards. So there must be somehow support for doing that. But if you look at it from a more broader perspective, I think, yeah, it's really just, it's dialogue forums that need to be put into place, and somehow the discussion over standards and their implementation in experiment or in simulation or in some usage scenario. That's how you avoid niche forming.
Yeah. So you're saying it's a sort of triangle, really, that you have the standard, you have the use of the standard, and you have the tools that encode the standard, and you need all three of those working together in order to be able to achieve the standard. What you can't have is just the standard in abstract, or the tools independently, or it all done without actually being applied in use. So you need all three in place.
Yeah, absolutely. I mean, some of these started off with SPML. If you look at the history, it started off from a community where the people that were developing the software were also the people doing the modeling and the science. But unfortunately, in most cases now, things are more specialized, where these are all different people. Right. And especially since everybody has different domains, and that you might find that there are more people oriented towards computer scientists working on the standard, on the technical sides of things. But then that could potentially lead to a detachment from the people that are actually making the models or and doing the experiments. So for me, there's as important, if you think of the systems biology cycle, there's an interlinked cycle on that which is running as modeling software and standards here.
Right.
Okay. Is that everything?
That is from me, Natalie.
Okay, we still have very shy people on our webinar today, so I'm afraid that's it for questions live, Brat, but it's been very interesting to hear your presentation and also to talk to you. Thank you very much for attending today.
Yeah, no, it's a pleasure. Great.
Okay, thank you very much. And as I said to everyone, you can ask questions later on the forum, and thanks for joining us today. We'll be running the webinar again next month. We're having a break next month because it's Easter. Then the month after, we'll be picking up again. So you'll get invites to that in due course. All right. Thanks very much.
Okay. Great. Thanks, Natalie.
Bye.
Bye.